Hey biology students, in this video we're going to discuss the central dogma of biology. In the last video we described the differences between DNA and RNA, both of which are nucleic acids, and comparing that to protein being built of something completely different than DNA and RNA, protein in a different category of macromolecules, and of course proteins, that is what makes up your phenotype. So recall, when you look in the mirror, you're not looking at your DNA, you're not looking at your RNA, you're looking at the product of your genes. The products of your genes are your proteins. That is your phenotype, that is your physical appearance, that is your physiological traits. So all of that is encoded by the DNA molecule. So in this video, we're going to discuss that process of how your genes are decoded within your cells to produce your phenotype, to produce your proteins. This process is referred to as the central dogma of biology. Also, another term we use is gene expression. So I have a definition here on this screen. Gene expression refers to how those genetic instructions within your DNA are decoded by cells to produce proteins, to produce your phenotype, your physical characteristics. So this is referred to as, like I said, the central dogma. Because there is a process, sort of a flow, if you will, of how the cell decodes genes. So it begins with the DNA molecule holding the instructions for your traits, and that DNA molecule is then copied into temporary molecules that we call RNA. Now, specifically, when we see that copying off of the DNA of specific genes, we call that mRNA for messenger RNA. And then that messenger RNA is used sort of as a blueprint to tell the cells what order of amino acids to add in the building of a specific protein. So it's this flow from DNA to mRNA to protein that is referred to as the central dogma. So in this video, we're going to describe the two steps that are involved in this process. In the last video, we already introduced the first step. So the first step is the copying step from DNA to mRNA. So when the DNA is copied, we call that step transcription. And remember, the word transcription has the word scribe in it. So think about a scribe or somebody copying a message over and over and over within the cell. And that is the job of the enzyme that's involved in the transcription process to act as the scribe. And the second step is what happens when the mRNA is used as a blueprint to tell the cells what protein to make. And that is the second stage, and that stage is called translation. And that word translation probably conjures up ideas of language translation, right? So if you're translating something from English to Spanish, you're changing languages. Well, that's actually what's happening in the cell in translation. If you notice this step, you're going to translate the language of messenger RNA, which recall that language, that chemical language is nucleic acid. It's written in the alphabet code of the mRNA, which is A, G, U, and C. But we're going to translate that code into a new language. The language of proteins is not the same as the language of nucleic acids, is it? What is the language of proteins? What are the proteins made of? That's right, they're made of amino acids. So we're going to be changing from a chemical language, once again, the alphabet of RNA, A, G, U, and C. Okay, now recall the alphabet of DNA was A, T, G, and C. So the alphabet is really similar between DNA and mRNA because they're both nucleic acids. But when we translate to protein, we're actually changing language. We're changing to the language of amino acids. So that is a different language. So in the last video, actually first, let's talk about an analogy. So I like this analogy when we're talking about the steps of the central dogma. This is sort of cute. So the concept is that DNA is like a cookbook. In other words, it is a giant instruction manual that the cell has with thousands of different recipes in it, where each recipe is basically a gene. Okay, so the DNA is composed of many chromosomes where we have many different gene recipes. And that cookbook, by the way, it in the context of how it works with DNA, that cookbook resides in the nucleus. And it never leaves the nucleus. So the DNA stays in the nucleus. The only thing that ever leaves the nucleus are these recipe copies. And these recipe copies are the messenger RNA. In this picture, we're seeing the DNA cookbook being photocopied. They actually have a picture of a photocopier here, and they're calling that, again, the transcription step. So we're transcribing the DNA by just copying a single recipe. In this recipe, they're saying we're going to copy the recipe for this protein cake, basically a chocolate cake, okay, is what I got pictured here. The idea is that the cookbook has a whole lot of other recipes, though, doesn't it? Maybe in that cookbook there's recipes for pizza, and there's recipes for cupcakes, and there's recipes for appetizers and desserts and all sorts of different things, right? In the same way that there's recipes for all of your specific physical traits that are in your DNA. So in the transcription step, we're only copying a collection of genes, or maybe even just a single gene, to make the mRNA. You can also make many copies of the same gene. So it doesn't have to be a single copy, but you can actually have many copies of that messenger RNA if we need a lot of that protein synthesized, for example. So then the analogy continues. You have this recipe, but that recipe is just a piece of paper, isn't it? If you go with the analogy, you photocopy from the cookbook, you just have this piece of paper, but we're not going to eat that, are we? That's not our luscious cake. So what do we need next? Well, we need somebody to assemble the ingredients that are written on that recipe form. And so here we have the remaining players of the story. We have the ribosome, also known as the chef. We have the tRNA, also known as the kitchen hand, somebody who's going to help out the chef. And then we have the ingredients. And remember, we're going to be building a protein here, so the ingredients have to be the amino acids because that is what proteins are made out of. So they didn't label it here, but of course, everything involved here on the right side of the arrow, this would all be the translation process. So this is going to occur where the ribosomes are, which would be either they're going to be floating in the cytoplasm, or remember there's attached ribosomes in the rough ER. So the job of that ribosome chef is going to be to use the instructions of the mRNA and assemble the correct ingredients in the right order. In the same way that when you bake a cake, if you've ever baked a cake for somebody, you've got to add those ingredients in the right order, don't you? You have to mix the wet ingredients first and then the dry ingredients and then mix it all together. And you've got to follow the actual steps of that recipe because otherwise you won't make the desired cake, will you? So assuming you follow all the recipe order correctly, you do end up with the correct assembled product at the end. So this is what I showed you in the last video. We already discussed the transcription process and we started with an example showing the DNA molecule where we only wrote out the sequence of those nucleotides. And then we showed that there's this copying step, the scribe, and the scribe being the RNA polymerase enzyme that copies the template strand of the DNA to create the mRNA. Now here's the figure from the textbook showing that same process. So they're showing us the DNA molecule in blue. They're showing us this big sort of beige circle. The beige circle, this is the enzyme. So they're showing you this RNA polymerase enzyme, and it's acting as a catalyst, of course, to open up or unzip the DNA and expose the template strand. So they've got the bottom strand here, what is shown in the 3' to 5' direction. This one is the template, and that is the strand that is used to create the complementary messenger RNA. So the messenger RNA is shown here in pink. Let's add a little M there. So that's the messenger RNA that's being transcribed from the template strand of the DNA molecule. In the last video, we also talked about tRNA. The analogy is that tRNA is like the kitchen hand. It's going to assist the chef, the ribosome, because the ribosome needs to add the correct ingredients, and the tRNA is going to recognize and bring in those correct ingredients. Now, I want to draw your attention to one other part of the tRNA that we did not discuss last video. It's the end that we call the anticodon. Let me go back a slide to here. Here, I need to give you another term to understand the mRNA. So every 3 letters of the mRNA, let's just put a box around every 3 letters of this mRNA, which, by the way, we made this really short. That's just for simplifying it for the video, but genes and transcripts made from genes are really long, actually. Most of them are going to be thousands of nucleotides in length. So we're just really simplifying it, guys. Okay, so every 3 letters, there's a term for this. Every 3 letters of the mRNA is referred to as a codon. So that's a codon, that's a codon, and that's a codon. So we have 3 codons here. Remember, it's every 3 letters. So a codon is defined as every 3, well, we should say nucleotides of mRNA. And it's always read from the 5' to 3' direction. So in the way we've written it here, we're moving from left to right and how those codons would be determined. Okay, so notice this term now on the tRNA, it's called the anticodon. So that means there's a region on the tRNA that binds complementary base pairs with the codon of the mRNA. So that's actually how the tRNA, so we'll write that here, so the anticodon binds to mRNA to an mRNA codon. So it's just one tRNA. Each tRNA recognizes each codon with its anticodon. So it has a complementary sequence that would bind there. That's how it knows which amino acid to bring in the correct order. The translation process is also referred to as protein synthesis. This is the step where we have a language change again. So we're moving from mRNA language, the nucleic acid language, to the protein language, which is the amino acid. And that is what happens in translation. Recall this occurs where the ribosomes are located in the cell. In this diagram from our book, we're seeing that picture displayed. Let me also show you how I would do that picture in my notes. So what they're showing you here is the ribosome, and they're showing you that the ribosome is really composed of two subunits. Sometimes I refer to these as hamburger buns, if you will, like we're making a sandwich. So I hope you're not hungry here as we go into all this food terminology. So hamburger buns, if you would, okay? So we have what's called the large, so it's called the large ribosomal subunit. And then we have the small ribosomal subunit. So the ribosome is composed of two subunits. And the ribosome itself is composed of protein and another type of RNA, which is called ribosomal RNA. So if you can imagine this top bun and this bottom bun of the hamburger, then we're making a sandwich. So what is in the middle? Well, you can see that pictured here in the diagram. That would be your mRNA. So the mRNA transcript that we made during transcription would be in between the two subunits, sort of like making a sandwich using the mRNA as the meat in the middle. So now we know that the mRNA is composed of codons. So every three, we'll just make them little zipper notches, okay? Every three notches on the mRNA, those are referred to as a codon. So let's circle the codon, okay? And of course, we've got a whole bunch of other codons as well, but let's show what happens. So the tRNA that matches that specific codon will come in and will actually bind. Let's make it a different color. So the codon, the anticodon of the tRNA, when I draw tRNAs, I usually just draw them like a little T and then with a little amino acid. So this is an amino acid here. So the amino acid is attached to that tRNA, and the tRNA comes in and binds. So it has its anticodon that matches a specific mRNA codon. And that's how it knows where to sit down, okay? And then it sort of scoots over, okay? And then we have another tRNA come in. So we get more tRNAs coming in, carrying an amino acid, and then we actually get everybody scooting down, and we get the tRNA that has lost its amino acid to exit. And what happens is you end up, every tRNA that comes in drops off its amino acid as it recognizes the codon, and then we get this long chain of amino acids that starts to form. We call it the polypeptide chain. Now this is a moving process, so it really helps to see this in action. Let's take a look at an animation of this. Okay, so I like this animation showing the process of translation. I'm just going to talk you through this one. So watch what's going to happen. Right now on the screen, we have in pink, they're showing us the mRNA transcript. And then in purple, we're about to see the two subunits of the ribosome come down and clamp down on the mRNA. So see those? So we've got the large subunit and the small subunit, like hamburger buns, right? And it sort of clamps down on it. And then here, this molecule is the tRNA. And just like in my drawing, we're showing that it does have an anticodon and it has the what looks like a little bead, but that is actually an amino acid that's attached to it. And so that's how it knows where to bind. So it sits down on it, and then there's sort of this process of what happens, because then the next tRNA comes in and recognizes the second codon, and then everybody shifts over. That's what's sort of hard for me to draw on the diagram we were doing, but see how everybody shifts to the left, and then this empty tRNA now leaves because he dropped off his amino acid, and that shifts to this tRNA temporarily until the next tRNA comes in with the next amino acid binding down onto the codon, and then everybody shifts over. And so there's always somebody in the ribosome that's facilitating the connections between the amino acids. So here comes more and more and more, and they just keep coming in and binding to the codon, bringing in the, remember they're called transfer RNA because their job is to transfer the amino acid to the ribosome. Finally, this process terminates or stops when a stop codon is read by the ribosome. And a stop codon basically tells the ribosome that we're done. We made the protein, so everything gets released. And now this is a sequence here of amino acids that we call the primary sequence of the protein. Remember, that's just the linear sequence, but it has to fold because proteins always fold into some complex shape. So how do we know which codon codes for which amino acid? Well, that is a Nobel Prize award as well. So the Nobel Prize, I believe in 1968, went to a team of researchers who decoded how codons work. And they did it in bacteria, but it actually is universal across all life. And this is what we call the genetic code table here. This is referred to as genetic code table. And it's not a table that we memorize. It's similar to, in chemistry, they have the periodic table of the elements. Nobody memorizes that. You just use it as a reference table, don't you? So you need to learn how to use the genetic code table as a reference table. So what this is showing us is it's showing us codons. So you're seeing you're seeing 3 letters of RNA language here. And then you're seeing that these codons code for specific amino acids. And those amino acids are shown here in 3-letter abbreviations because they do have longer names. Like this one is, the abbreviation is CYS, that's the 3-letter abbreviation. This is actually cysteine. So CYS is actually cysteine. That's the full name of that amino acid. And TRP. Tryptophan. Anyways, it's not essential that you know the full names at this level in biology, but you should know how to decode the mRNA and write in the correct amino acid. So let's practice that. So I just copied the mRNA that we've been working with in this video, which we only had 3 codons, recall. So we just simplified it. We just have these 3 codons to deal with. And what we want to do is we want to translate it. So we want to translate it, and we're going to translate it by writing in the sequence, the correct sequence. Remember, the sequence matters. It's just like baking a cake where you have to add the ingredients in the correct order. Otherwise, the protein or the cake is not this correct cake. So that order is really important in making a protein. So what we do is we go and look at the codon table, the reference table, to figure out what amino acid is coded for by each of these codons. So the first one is ACG. Here it is. I'm going to circle it. So there's ACG, codes for this amino acid. THR is the abbreviation. The full name is threonine. THR is what I would write. And then we always just make a little dash because this is actually going to be a type of covalent bond between these amino acids. That's what's going to happen. Okay, UCA. Here it is. UCA codes for serine or SER. Dash. GCA. Here it is. GCA codes for the amino acid alanine or ALA. There we go. So that's all there is to it. You have to be provided with the genetic code table, but you need to know how to learn how to use it. Now, we did a really simple example here. Notice that there's a little bit more to this. One is that there is something called a start codon. This is the AUG. A lot of times the AUG is the first codon in a protein. And then usually it also has a stop codon as the last codon, so the ribosome knows to be done. I want to draw your attention to two more important things about the genetic code table before we wrap up this video. The first thing I'd like you to realize is that we use some descriptive words regarding the genetic code. So the genetic code is redundant. And what we mean by that word is, you can see, I've already written on the screen, but it says there's more than one codon can code for each amino acid. So if we look at, let's say, the amino acid serine, for example, SER, serine can be coded for by UCU, UCC, UCA, UCG, and look over here, also AGU and AGC. In other words, again, there's multiple codons that code for the same amino acid. That's what it means to be redundant. There's codons that are doing the same thing in the cell. And that's really to allow for mutations. So when there are mutations, we'll talk about that in the next video, but mutations can be harmful to the cell. But if a mutation doesn't change the amino acid sequence, then it's not harmful. Okay, the next important term that you should know in describing the genetic code is that it is universal. Remember I said that the scientists who described first the genetic code, they did their lab research in bacteria. But it's a code that applies nearly universal across all life forms on Earth. And that is because all life on Earth is related. So there is a common ancestor, a single-celled ancestor to all life on this planet. And there's a common genetic language that links all of us. So the same sequence of RNA codons in a bacteria, in a plant, in a yeast, in a human cell will be decoded in the same way, nearly the same way, and use the same codon chart. So this chart can be used for any organism, nearly every organism on Earth. Okay, and that's going to be important to understanding why biotechnology works, why genetic engineering works, why we can actually add or remove genes from organisms in a lab process called genetic engineering. That can happen because of how the genetic code is universal across all life. Okay, so that's all for this video, and I'll talk to you later.